Thanks.
It will take some time. Yeah.
Okay, should I
Yes,
just
yes, you can start. Yeah, it started.
okay.
Yeah.
Okay, so right, so this idea that one should not concentrate on the details of the microscopics is, of course, familiar to us in condensed matter systems, most famously from Anderson's paper on more is different. So this is sort of the guiding philosophy behind what I'll be talking about, and so I'll just jump in and tell you about the particular system that I'm looking at this in. And so I'm looking at developmental biology, and developmental biology is a field which has had a somewhat more theoretical a history of theoretical and conceptual thought than many other fields of biology, for example, molecular biology and so on. And so I'll tell you a little bit about how developmental biologists very briefly think about embryonic development. I'll tell you about the idea of Waddington landscapes. And then for most of the talk, I'll focus on how one can sort of mathematically translate some of these ideas and do this in a way that I think is both illuminating but is also at perhaps a more practical level useful for fitting experiments in developmental biology. So to start off, this is sort of very, brief introduction to the idea of cell fate, which in development, right, you start off with a single cell. And of course, this is many people have been fascinated by the fact that you can go from a single cell to a fully developed organism. And the way that early developmental biologists would would think about this is they would basically say that you can take an early embryo and you can mark different regions of that embryo, and then as it develops you can see what happens to those regions, and so you can sort of see what fate different regions in the embryo end up taking. So initially, of course, you just have a single cell, and then in early stages you have something that's very homogeneous, and then slowly and slowly you get these symmetry breaking transitions, and eventually you have something that's of course very heterogeneous and fully developed organism. uh, this is of course not quite accurate, because in most complex organisms the cells are moving around and so on, and so the regions don't really stay fixed. But in the case of some organisms, in particular the worms, so C. elegans in particular, you can really make a very good kind of fate map where you can there are about 1,000 cells in the worm, and you can really, as these cells develop, you can sort of say what fate each cell will end up taking. And so that's the figure on the bottom. And of course all these fates have names which one doesn't need to worry about, but this is just to say that cell fate is a concept that sort of goes back to very early experimental developmental biology. Now, this is at the level of the embryo, where you're having seeing something complex happen, but really at the level of the cell which is deciding what fate to take. For example, you could think eventually cell either become a skin cell, or it'll become a neuron, or whatever other cells, many cell types are there in the body. At the level of the single cell, what is happening, you could argue, is even more complicated. At the level of the embryo, of course, things are complicated, but at the level of a single cell, you have what are called gene regulatory networks, which basically are telling you how, when an external signal is presented to the cell, which is often in the context of development called a morphogen, what what happens inside the cell, sort of what chemical reactions, what genes are turned on, and so on, what proteins are expressed. So what happens inside the cell that makes it change its chemical and of course mechanical properties. And usually the way that people think about these gene regulatory networks, which are essential for modeling the decisions that the cell takes and the kinds of fates that the cell ends up taking, is by really looking, or trying to look at the chemistry of all of the things that are involved. And so these are proteins, there's some transcription factors that activate things and so on. the details one doesn't need to worry about. But at the level of equations, which equations are of course not, these are not fundamental laws, these are phenomenological equations, which one is familiar with from chemistry, like Michaelis Menten and so on. At the level of these equations, you can write down basically an equation for each chemical component that you're interested in inside the cell. And you can try and write down a set of differential equations which tell you how the chemistry of the cell will change. And the issue with that is, of course, that even in simple, relatively simple cases, so on the left here is a diagram of cells in rats where they have to decide between either dividing or differentiating into a particular kind of neuron like cell. Again, they're just two decisions that they have to take. And on the right is sort of a model of all the genes and all the proteins and so on that people think are involved. And then you can write down these differential equations of how what things are activated and so on. And it's purposefully, I'm not showing you the details of the equations, but only if you zoom in into one of them, and you can sort of see that all of these equations have these various parameters. There are these threshold coefficients, there are these rates of reactions and so on. And all of these parameters are typically not very well measured. And in fact, most people would not really go bother to measure them. And so you have sort of models of phenomena which are quite from a perspective of a physicist, they're not very nice, in the sense that they're basically over parameterized. And the fact that these models are over parameterized, actually you can make this into a sort of precise statement and understand this using information theory and so on, which I won't go into today. But this is just to say that uh, in biology often has sort of over parameterized models. uh, and one is interested in the spirit of condensed matter physics to try and figure out how to simplify them, how to come up with effective theories, maybe at different scales or different time scales or so on, which capture some simple dynamics that you might be interested in. Okay, so now even though from, I guess, in condensed matter physics, this is a relatively new idea, going which was really concretized with the renormalization group and so on. In the field of developmental biology, there was because a lot of the conceptual things were actually done before one knew too much about the molecules involved, too much about the genes. There was what can, I guess, only be called as a more emergent description of what is happening in development. And so the idea was that, as I showed you, at the level of the single cell, you have this complicated chemistry happening, and the cells are, as the chemistry is changing, the particular fate the cells are taking will change. And Waddington, who was a developmental biologist, he came up with this famous metaphor of a landscape. And it was by this, he illustrated this with the picture, which has now become quite common, where you have a ball which will roll down some hill, and then you have some minima at the end, some stable points at the end, where the ball will eventually stop. And these different minima correspond to different stable fates that the cell can take. And of course, Waddington was interested in this because he was seeing, as a developmental biologist, that the outcomes of development um, are very robust. And despite sort of all the things that could go wrong, you typically very robustly develop into a whole organism. And one sort of simple example of that is the fact that you have 5 digits, as they're called, or 4 fingers and a thumb in your hand. And it's quite rare that you see someone with 6 or 4. So on. So he was seeing the fact that development is quite robust,
So,
and yeah,
can I interrupt?
yes,
This landscape that you kind of show it. So what is this landscape? What you are plotting? I mean, is it like
right,
some energy, or what is the y-axis in some sense?
right. No, so this is the question, and Waddington didn't really answer this question, and from so maybe I'll just go into this. This question sort of gets me into the next slide, and so the idea is, I guess, I mean, I could even stay here. The idea is that, of course, in the in the chemistry that I was showing, these various kind of concentrations of different chemicals, you are in some very high dimensional space, which you can parameterize with as many number of variables as you think are relevant to the problem. And the um, view that I think one uses to translate what Waddington was saying is that from this high dimension there is some dynamics in this high dimensional space, and that dynamics really can be projected down to a lower dimensional space, where sort of the ball is effectively rolling down into different fates. And how that projection happens, and what that lower dimensional space is in terms of any physical observables, is a much more complicated question. But it's essentially saying that the actual space is very high dimensional. There's a lot of chemistry going on, which I showed you in the previous slide. But effectively the dynamics that you're interested in from the point of view of the cell is happening on a much lower dimensional manifold. And there's some the way one could think of it from a point of view of a dynamical systems is to take a very simple example. If you had, let's say, only two chemical components you were interested in, one gene or whatever else, some protein which was auto-inducing, which induced the production of itself and had some decay rate, and something else which was regulating the production of this first protein or first gene. I mean, these are proteins are what you're really interested in, but the genes are regulated and then lead to the expression of a protein. And so then in this two-dimensional space, you can think of these equations as, of course, making a dynamical system, and you can plot the flow lines. And so if you have regulator and product, you can choose parameters in a way that you get two stable fixed points and a saddle point in between. And this was basically worked out by a student of Waddington. This way of looking at the fact that dynamical systems theory is the right mathematical tool to think about development was worked out by a student of Waddington called Slack. And he realized that you could take some of the terms that were being used in development, like a cell is differentiated when it adopts a certain kind of fate, and relate them to dynamical systems theory, that you have a particular fixed point, or it's there are other terms in development. A cell is determined when, despite external signals, it will still go to the fixed point. So you can imagine there's some small region around a stable fixed point where things will flow into it even if you have some external signals that are entering into these equations, which here are not modeled. And so the idea is that you have some high dimensional space, but the effective dynamics can be reduced to some lower dimensional space, which in this case I'm showing as some two quantities that you might be measuring or interested in. Yeah.
So are you effectively saying that the outcomes of the developmental process are represented by the fixed points?
Yeah, correct. Yes. Yeah. The fixed points are the various different outcomes you could take.
Yeah.
And these things in between are saddle points, which are the points at which the cell hasn't decided what particular fate it wants to take.
Yeah. Okay. Yeah. Okay. Yeah.
Okay.
Thanks.
So and this idea of using dynamical systems, in contemporary times has sort of come in, and now of course you have the advantage that you can use sort of your experiments or the kind of data that you can collect has become you're able to collect better and better kind of data, and so you're really interested in modeling these developmental trajectories, and so the idea that you can use such dynamical systems to model these developmental trajectories sort of recently again come to the fore. At that point when it was developed, it was not really used mainly because it was hard to most of the kinds of experiments that were done didn't really examine these trajectories in any kind of detail. So the basic idea, which really goes back to René Thom, is that these Waddington landscapes are linked to what are called Morse-Smale systems. And I won't go into the technical details of Morse-Smale systems. These are called structurally stable systems. But the idea is sort of that if you have a vector field which only has saddle points and stable fixed points, and you could have unstable fixed points and so on. So if you have some arbitrary vector field where you get rid of some cases like chaos, and actually technically periodicity is allowed, but for the purposes of this talk one can ignore periodicity also. But if you have systems where you basically have saddle points and stable and unstable fixed points, if they're produced by some arbitrary vector field, it turns out that there's a theorem that you can always write down this vector field as the gradient of a metric times, sorry, a gradient of a potential times a metric. And so René Thom sort of saw the link of Waddington landscapes to more Smale systems. As far as I'm aware, he wasn't aware of this theorem that Smale also proved, or maybe if he was aware, he didn't particularly apply it. That these more Smale systems can always be written as a gradient of a potential times a metric. And this is, of course, interesting because now this means that you can, this landscape that was being discussed earlier, one could really mathematically say that there is a landscape like that there is some function whose gradient will give you the correct dynamics. But the caveat is that it's not always just the gradient of a function, but you have to put in an additional factor, which is this metric. So this sort of inspired, are looking at different biological systems in development and seeing if one can really take this metaphor of Waddington landscapes and convert it into a mathematical statement about how to model these developmental trajectories as the gradient of a potential. And then second, of course, whether this kind of a model is a more parsimonious representation of what is happening in development as compared to the kinds of gene network models that I showed you before. Okay, so yeah.
Sorry, but if you just tell in biological systems language, what are these variables excise?
Right. I mean, so these are basically genes. I mean, this is what you're interested in ultimately. But these variables xi, one should really think of them as sort of abstract variables, which, as I said, are obtained as some projection from
Okay.
from gene activities. So I'm only right now. So this is actually development in itself. I should mention. Thanks for bringing this up. Development involves a lot of things. It involves changing shape, right? Cells change shape and so on, which is usually called morphogenesis. But for the purpose of this talk, I'm really only looking at the chemistry, which is basically determined by the activity of the genes. Which genes are regulated, which proteins sort of diffuse and send signals to other cells, and then regulate other genes and so on. And the gene regulation decides to a large extent the kind of fate that the cells end up taking. So I'm only focusing on the chemistry, but actually, of course, there are many other processes involved, which I'm not considering. Yeah.
Thank you.
So essentially, this is a way to solve this set of equations which you had shown in a few slides back.
Yeah.
So this is a way of converting those equations into this function f or whatever.
Right. So one could take that approach and actually directly try to take those equations and construct a function f, or one could believe that a function f exists and then try to construct the simplest, simplest potential which captures the phenomena that you're interested in.
And so, are we trying to construct it globally, or because only few variables may be relevant locally? So, are we sort of doing it locally, or do we construct this globally? Or?
Yeah, right. No, so okay. So that's a good. So here's the how you would want to construct it. So you have some. You take some arbitrary vector field. This is actually a system that not that I'll be interested in later in the talk, which is an activator and inhibitor kind of system. So now this is again in 2 dimensions, and The vector field globally here is shown in green, the flow lines of the vector field. So as you can see, there are two stable fixed points. There's a saddle point in between. And the question is, can you take this vector field and can you transform it into a metric times a potential? And this one can do in this case, in his example. So I've constructed a potential here, and the contours of the potential are shown in red, and the normals to the contours which determine the flow are shown as these red arrows. And then the role of the metric is to, at each one of these points, to rotate the red arrows into the green arrows. The metric is basically responsible for fixing the dynamical difference um, between the potential and the actual vector field. So what the potential has to have is the location of the fixed points. So really the fact that you have a few number of fixed points, which again, this idea goes back to the fact that in development, when you for example, make mutations or other perturbations in the system, you don't usually get new types of cells. You just get permutations of cells take different fates and they just permute which cells take one fate or the other fates. So perturbations and so on don't change the qualitative nature of the system. So that qualitative nature, that there are a few fixed points, this you want to capture in the potential. But then there's a dynamics when the cell, in this abstract space, hasn't decided what fate is going to take, and it's maybe at the saddle point. There's a dynamics that takes it from the saddle point to its eventual fate. And that dynamics, to really capture globally, you might need the metric. And it might not just be captured in the potential. So the potential kind of captures the topology of the system, and the metric can be used to fix the dynamics if it doesn't match. Okay, so you're not just, in development, you're not just interested, of course, in the system and the kinds of fates that cells can take, and, you're also interested in perturbing the system, right? And what developmental biologists do is they'll do mutations and then they'll see what phenotypic effects they have. Or actually, Waddington, what he was doing was, and why he came up with this landscape, was he would take these flies and he would give them a heat shock. And then when there was a heat shock, then the phenotype of the flies would develop a second pair of wings. So when you have these external perturbations, it changes the kinds of phenotypes that your cells take. So you're interested in seeing how, given some external perturbations to these vector fields, how that will change their qualitative behavior. And of course, this is what is done in bifurcation theory. And again, to take a very simple example, if you take a one-dimensional equation which has some autoinduction and some degradation, so the autoinduction here is shown as the orange curve, the degradation is a straight line in blue. When you have some external inducer, the simplest way to model it is some constant into these equations. It's changing the production of this protein. So when you have no external inducer, then you can have two fixed points, which are given by the point where the straight line intersects with this curve in orange. But as you change the level of some external inducer, that can change the level of fixed points you have. And this is, of course, standard bifurcation theory, which you are interested in here to see not just what a developmental system is like, but what happens when you perturb it. And bifurcation theory, of course, has examples of several different kinds of bifurcations that you have. The key insight, again, is the first to remember was that you're dealing with some more Smale systems, which you can write as potentials and so on. The second sort of key insight is that there are only two kinds of bifurcations that are generic. And generic is a mathematical term, that has a particular definition, but again, one can really think of it as, if I take a set of equations and I perturb them, I put some arbitrary perturbation on them mathematically, does that change the qualitative nature of the bifurcation? And it turns out that there are really only two bifurcations that are generic. and they are what is called the saddle node bifurcation and something else that's called the heteroclinic flip. And so to demonstrate this, so the saddle node is here in case. So saddle node bifurcation, here I have 3 stable fixed points and I have 2 saddle points in between. And the saddle node bifurcation is basically when a saddle point and a fixed stable fixed point annihilate each other, which is shown in this sequence of plots. And this happens as I change some external parameter. So this is an example of a generic bifurcation, and the idea is that if I've of course made this set of plots with a certain set of equations. If I make some small perturbation of any arbitrary kind to these equations, that should not change the fact that I get a saddle node bifurcation. Whereas in other kinds of bifurcations, for example, pitchfork or other kinds of bifurcations, if you make small arbitrary perturbations to them, that changes the character of the bifurcation itself. So they are not, in that sense, generic bifurcations. And this idea of genericity is quite interesting. It again says that the qualitative nature of the system doesn't change given arbitrary perturbations, and it's sort of different from, I think, what one is used to in physics, where one is really used to symmetries. And the idea that there's some special some symmetries, which basically, once you know the symmetries, you determine the theory. And genericity is basically sort of, in some sense, the opposite idea, that there are no special symmetries in the system. And I would say that a lot of the study of biology has shown that typically biology doesn't really the way biological mechanisms works are through heterogeneity, are through lots of tinkering and so on, and not really through preserving some special symmetries of the system, particularly at the level of gene regulation. So saddle node bifurcations and heteroclinic flip, this allows you to, so heteroclinic flip also, let me show you. So heteroclinic flip basically says, yeah, again, I take the same figure where I have 3 stable fixed points and 2 saddles. The idea is when I exit this saddle, so if I am at some point which is on the outset of this saddle, which sort of is unstable, which stable fixed point will I end up in? And as you can see on the middle plot here, if I'm at the outset of the saddle, I'll end up at this top fixed point. Whereas on the bottom plot here, if I'm at the outset of this saddle, I'll end up at the bottom fixed point. And of course, there's some point in between. Again, these plots are obtained for different values of parameters and the equations that I have. And there's some point where these saddles sort of align exactly, and you have a totally, this outset of the saddle actually goes into the inset of this second saddle. And the heteroclinic flip is saying that you can change basically the global connections of whether this fixed point is connected to this one or whether this fixed point is connected to this one by changing the relative positions of these 2 saddle points. And so these 2 kinds of bifurcations now, if you consider, as I said, I was changing some external parameters to produce these plots. If now I consider a space of 2 external parameters, let's just call them P and Q, there's a line in this 2 parameter space where I get these 2 kinds of bifurcation. So this line, which is shown here, is where I get the saddle node, where I go from a 3 fixed point system to a 2 stable fixed point system. This line here is where I get the heteroclinic flip, where I go from this kind of, this fixed point being connected to this one, to this fixed point being connected to this one. And it would seem like, at this point, I mean, why is one spending so much time in, doing, sort of working out these details of these dynamical systems? And I'd like to show you later on that actually I think these things have sort of strong experimental consequences. But for now, I will just say that what now you've done to sort of summarize is that you knew that you had some large dimensional system at the level of gene regulation. But at the level of phenotypes, so all these experiments in developmental biology have shown you over the years that your system is not too complex, that there are only a few fates your cells can take at particular stages in development. By sort of ignoring any knowledge, without using sort of any knowledge about the nature of the equations, which in any case, one doesn't really know the microscopic nature of, the chemistry in vivo in these cells. at the level of the phenotypes, at the level of what fates the cell is taking, one can already make some claims about what kinds of transitions are expected if you perturb things. And this is exactly the kinds of experiments that one is interested in developmental biology. You make perturbations, you make mutations, you put external factors, you change the environment, and you see how that changes the phenotype. So in that sense, one, that's why the idea is to go from microscopics to phenomenology. One is, I haven't shown how to go from the microscopics to the phenomenology, but to the more coarse-grained description. But this is to say that one expects from the experiments that a coarse-grained description exists, and then one is interested in parameterizing it. Yeah.
But in case of developmental biology, I thought you don't have a sense of what the alternative phenotypes could be like unless you do the experiment.
Right.
Isn't that true?
I mean, what exactly do you mean? Alternative phenotypes?
Alternative phenotypes in the sense that if you perturb the system by giving some sort of heat shock, for instance,
Yeah.
you really know that the eventual developmental trajectory will end up in producing two wings, two pairs of wings, right? Instead of one. Or would it end up in some other phenotype?
Right. Right. So I should make a distinction here. So far I've been sort of talking, and this is actually the Waddington landscape is used both at the level of a macroscopic phenotype and at the level of a single cell. So far I've sort of been talking at the level of different fates that a cell can take, and then later on I will talk a little bit about spatial pattern formation, and at the level of the phenotype as a whole. The fact that you get two wings is of course quite a big change. Whereas here, for now, one can think at the level of single cells at different stages of development. It's quite well characterized what different fates the cell can take. And the idea behind this parameterization is that, for example, at the beginning, you get this specification into mesoderm, endoderm, and ectoderm, these three germ layers, and this is sort of well known and across different species. So there are three different fates that the cell can take. Even before that, there are transitions, which I'll give you examples of. So the fact that these fates exist is sort of well known, and the idea is that by parameterizing the dynamics in a simple way, you can now not just look at these static fates, but really look at development as a dynamic process, and the fact that at different stages in development, different kinds of perturbations will produce different results.
Okay, so what you are saying is with this method, you can effectively predict what kind of alternative developmental trajectory the cell might take given a certain perturbation.
Right. Given that you sort of know the kinds of things that can happen.
That can happen,
yeah.
right?
Yeah.
Okay.
Yeah.
Okay.
So the fact that does it really make a difference? Since could you have a second order differential equation and time also, which this would go through for that also, right? Or
right. I mean, second order is two first order. I mean, two first order differential equations in time. So yeah, in that sense, one is not.
Because the kind of modes which you get are different, of course, for second order equations and first order equations. So
right. So what this is relying on is this idea of structural stability, really, which is basically that the fixed point structure doesn't is sort of stable to perturbations, so for any sets of equations for which so again oscillations here one is not considering. So again, that's something that you can get in second order things and also in first order things, but one is not considering oscillations. One is just considering things where you have some stable fixed points or saddle points and so on.
So basically at one level, so of course at one level one can think of this being like a Lagrangian framework as opposed to Newton's equation of motion framework, or of course in some sense it is more. So it would be nice if you could just elaborate in what ways it is more, of course.
Right. Well, I mean, that's an interesting analogy. It's I mean, I would say that the um, more than so, I mean, Lagrangian and Newtonian there are sort of mathematically equivalent ways of looking at the, or roughly equivalent ways of looking at the same system. Whereas here, the idea is that you're also interested in dimensional reduction, which is not really something that. So you're interested in dimensional reduction basically because you're interested in models that don't over parameterize. And
that's not something which that's not something which you've discussed here. So that is the general program, but at least the prescription which you've discussed here,
yeah,
where
yeah.
does dimensional reduction come in there? Or
Right. So that's an interesting. I mean, so the dimensional reduction. Well, one is of course that there is some dimensional reduction in the sense that at the level of phenotypes you don't see too much. Here there is a claim about dimensional reduction in the sense that you can say that if I am interested in looking at sort of only these. The dynamics on this phase space with a few fixed points and a few saddle.
okay, the directions are sort of fixed by the gradients or whatever.
I can sort of ignore
Right.
things that are happening in other dimensions. This is the idea, and which, of course, as I said, there's some large dimensional manifold, and this is some cross section of it where the relevant dynamics is happening. But the dimensional reduction claim is that you can ignore sort of the other, all of the other dimensions that are inherently in the problem.
Okay. So,
Right. I mean, so there is a more difficult question which I'm not addressing, which is how do I go from microscopic equations and coarse grain and produce these equations? This I'm not addressing. This is, I think, a pretty difficult question in biology. How do you coarse grain at all? Which is why I'm sort of calling it phenomenology, where you're saying that there's a smaller dimensional space in which your dynamics is happening. The question is, can you parameterize it in the simplest possible way, and whether you can understand something about the kinds of dynamics that can happen without knowing pretty much anything about the microscopic equations whatsoever, which is why this idea of genericity and so on.
But you are still looking at some of the relevant variables that probably went into building the microscopic picture, right?
Correct.
And so how do you identify what those relevant variables are? What subset of the microscopic set of variables would constitute these set of relevant variables?
Right. Yeah. So this is there's no the reason that so those variables are sort of identified for my purposes by the biological experiments. There's a question: how do you go from the knowing the microscopics to actually constructing the relevant variables? This I'm not addressing, but the biological experiments will typically vary two things which they know matter. It's some protein that matters for the decision. We're going to vary the expression of that, so that can be one of the parameters, and then some other pathways involved, you can vary the expression of that. The fact that the problem is tractable at all from a biological perspective, I'm sort of using to say that you have a few relevant variables, which is what you're perturbing. How they emerge, again, right, like in the renormalization group, how they emerge from the microscopics is, I think, a more difficult question.
Okay.
A question I'm independently interested in, but yeah, not something I'm addressing here.
Yeah, thanks.
Yeah. Okay, so so as I said, so there are these boundaries you can make in this two-dimensional parameter space, which tell you where the how these bifurcations happen, whether the flip and the saddle node, and the basic sort of result is that you can make an enumeration of the sorts of transitions that can happen if you have three stable fixed points. If you have three stable fixed points and you are allowed saddle node bifurcations and heteroclinic flips, then whatever the nature of the global vector field, in a space of two parameters that are in those vector fields, as you vary them, you can sort of enumerate the different kinds of, just globally, the different kinds of behaviors that you will get. And this is, I think, quite a powerful result, because it says that you can And again, 3 stable fixed points appear many times in these biological systems. And it says that you can take any one of them and you can make whatever model you like of them. You can take the genes, you can write down some chemical equations. If you vary 2 of the parameters in those models, like 2 of the proteins or whatever, you will generate one of these diagrams, which I won't go too much into the details of these diagrams, but basically the idea is that these curves, as I showed you, of bifurcations, can meet each other in 2 ways. Either they can meet each other in a parallel way, and then they form something called a cusp, or they can meet each other transversely. And just by sort of having these simple rules of how these curves can meet each other, you can sort of generate the diagrams that are possible for how a system of 3 fixed points can transition or can have bifurcations to monostable or bistable systems. And so let me just show you a few examples just to make this a little bit more concrete. And one of them comes from the worm, which is C. elegans. And there's a model of the development of the vulva in the worm, which has biologically been very intensely studied. People know at a certain stage that there are sort of 6 cells. These 6 cells are receiving a signal from a different cell, which is called the anchor cell. The anchor cell produces a protein, which is called the EGF, the epidermal growth factor. And then it sends this signal to these 6 cells. The cells also talk to each other using something called notch delta. Notch delta system, where these cells produce some again, they produce some protein, this goes some ligand, there's a receptor for it in the other cell, and so on. And so people know that EGF and notch are sort of the 2 things that are involved here. There are various experiments over 25 years of trying to vary the levels of EGF and notch and seeing how the fates that these cells change. It turns out these cells can take 3 different types of fates, which are given these names, which are not very illuminating, but anyway, P5.P. But they're shown here in colors, blue, green, and red. And so again, so now you have a dynamical system which has 3 fixed points. You have 2 parameters which you can control where a cell in this state space will end up, and you can ask what kind of which one of these diagrams it is more like. This was sort of, the geometric model was worked out earlier, but now we can anyway, we're able to see that it actually belongs to this thing called the elliptic umbilic. But let me give you a better example, which is this mouse, which is example from the development in mouse blastocyst. So this is very early stages of development in the mouse, and it turns out that you get 2. So first there's something called cleavage, and you get these things called blastomers, and then there are 2 differentiation steps. At the first step, what's called a blastomer differentiates either into something called the trophectoderm and the inner cell mass. And the second binary decision is the inner cell mass becoming either the primitive endoderm or the epiblast. The epiblast ends up becoming the embryo proper. It gets implanted into the uterus and so on. The primitive endoderm is sort of supporting tissues. So this is very early stages of development, about 3.5 days after fertilization, where you have this decision where the inner cell mass is deciding whether to become a primitive endoderm or an epiblast. And again, these colors are sort of shown here, how different cells end up taking one or the other fate. Now, sort of the question is, again, how does one look at this? And so from, one can look into the genes that are involved, and turns out something, again, there's some genes that are involved, some external protein called FGF. Depending on the level of that protein, you take the decision whether to become primitive endoderm or epiblast. And so people have some ideas about what this genetic circuitry might be. These are not confirmed, but you can make a genetic circuit and you can make some model of it. So this kind of a model was sort of made in this paper that's referenced, and turns out that if you look at this model in the space of 2 of the parameters of the model, in this case, one can choose the level of the FGF and some mutual inhibition between these 2 genes. The particular parameter you choose, again, doesn't matter so much. it turns out that the geometry of this model falls into one of the classes that we had, and it's, this class is called the dual cusp. Oh, I'm sorry, I think I missed, sorry, I was supposed to have something else here, but the maybe it's an older version or something, but this is now to try to show you that actually whether the geometry is the dual cusp, whether in the space of 2 parameters you have this kind of geometry or some other kind of geometry, can actually be quite relevant. In particular, right, as I told you, you have 2 fates, primitive endoderm and epiblast, and they're controlled by the level of this external protein FGF. Now here, if you had this type of geometry that's in the model, so these lines are bifurcation lines, where in this region you have just epiblasts being stable, in this region you just have primitive endoderm, this is a tri-stable region, and these are 3 bistable regions. The black line here is the particular parameter value for which this model was solved. But if you have this particular kind of geometry, this means that as you increase the level of FGF in your system, and you want to go from epiblast to primitive endoderm. So for example, if I took the mouse blastocyst, in vitro, and I put in a lot of FGF protein externally, I would find that all of my cells become primitive endoderm. You don't get half of them becoming epiblast, half of them becoming a primitive endoderm, PRE. But what this is saying is that as I increase the level of FGF, I will first have to go through a tri-stable region where I have 3 stable fixed points, and then eventually I will have to go to the primitive endoderm. Now, this is not the only geometry which is possible. Depending on how these bifurcation lines meet each other, I could go directly from the epiblast to the primitive endoderm without having to go through a tri-stable region. So I had a figure to sort of illustrate how this geometry could look, in terms of the flow lines. But I don't seem to have somehow missed it. But actually, the idea is sort of shown here. So one possible geometry is that I have this thing like an heteroclinic flip, where I have an initial state, this inner cell mass, and then I have epiblast and primitive endoderm. It's clear here that the primitive endoderm and the epiblast, these 2 stable states are connected to each other. I can transition from this state to this state just by having some sort of saddle node bifurcation here. I don't need to go through the 3rd state to transition from this state to this state. But if you imagine now instead a 1-dimensional potential, which also has 3 stable states, this is clear, this is 2-dimensional. In a 1-dimensional potential which has 3 stable states, to go from the left to the right, you sort of manifestly need to go to the center. Essentially, this has sort of an experimental consequence. As you vary the levels of the protein at different dynamic time points, you will either go through the center or you will not go through the center. And so this is to show an example of the fact that these geometries actually have content. And so I'm now working with some experimental collaborators to try and verify which one of these geometries actually fits this system better. So maybe I'll stop here if there are any questions, because I'm going to transition to spatial pattern formation. Okay, so I don't know, I guess I'm sort of a little out of time. How much longer should I go for?
Yeah, we asked quite a few questions, so I don't know, maybe another 10 minutes. 10.
Okay, 10 minutes is good. Yeah, so I'll just go through a little bit of spatial pattern formation, because of course you're not just interested in these systems with few cells, like I was showing you early stages of the mouse is about 32 cells. The vulva, you're really just looking at 6 cells. And so there you can look at these diagrams at the level of individual cells and how they're changing and so on. But you're interested ultimately in development in spatial pattern formation. And the classic model for how pattern formation occurs in space is there are 2 models actually. One is called positional information, which I won't cover. The other is this Turing mechanism, which is this idea of self-organized pattern formation, that you can have this breaking of homogeneous spatial symmetry and sort of spontaneously form these kinds of spatial patterns. And this has been quite successful in capturing various different patterns. For example, in in these seashells and these fish, and the left here is the actual biological thing, and on the right here is the computer simulation. So as you can see, this kind of a model with reaction diffusion equations has been quite successful in capturing the kinds of patterns that you see, particularly outside, zebras and so on. But the so here's an example of how it works in something which is called the Gierer Meinhardt model. Now, if you have something that's an activator and has an activator and an inhibitor, so the activator activates the production of itself and the production of the inhibitor, and the inhibitor inhibits the production of the activator. So you can write down a set of equations which have local activation and long-range inhibition, and often these kinds of equations are the ones that are used for these patterns. The issue is, of course, that from a biological point of view, it's actually quite difficult to identify any molecular components which correspond to the activator or the inhibitor. This has been done in very few systems. And recently there's been some work in saying, can one extend this to larger networks and still see these kind of Turing patterns? But these spatial patterns in Turing are actually quite hard to get. They're not easy to get, and this is why this activator inhibitor system became so popular, because typical connections, of activation or otherwise between two kinds of things won't produce spatial patterns. They'll just produce a homogeneous state by adding diffusion. So we were interested in seeing if one can look at what is happening in spatial pattern formation at a geometric level. So now the problem is a little bit different. You're not just looking at the dynamical system at the level of a single cell. Now you're saying there's cells that are talking to each other, and if you have some n cells, that you have some n-dimensional system. What is happening in that high dimensional? Again, now this high dimensional because also there are a lot of number of cells. What is happening in that high dimensional system to give you a particular spatial pattern, and can you understand this also geometrically? And so in this case, we took the simplest example. In one dimension, you take something like this, an activator inhibitor system, and you get pattern state, which in this case is just a bump. Your periodic boundary conditions, you just get some bump here, and the position where the bump happens, it depends on the initial condition. So it is right now at the center, but it could be anywhere else here. The peak of the bump, I mean. And so in blue is the activator, red is the inhibitor, and again, this is typical of the kind of behavior that you see in these systems. Immediately, if you look at it, then it's not obviously a model which is potential. In particular, these equations that I showed you, if you take the gradient, right, if you look at the cross derivatives, then it's, the cross derivatives don't match. And in fact, I won't show you, but if you can believe me that it's quite hard to write down a local potential of two fields where one acts as an activator and one acts as an inhibitor. Basically, this condition of activation inhibition means that the cross derivatives have opposite signs. And so inherently, it's sort of something with a curl. It's not potential. So we realize that actually this is these are sort of the wrong, looking at spatial fields A of x and h of x and trying to construct some potential in them to find out the geometry is the wrong way to look at it. And instead, we started by looking at sort of very simple discrete examples. So if you just have two cells, one can be high, one can be low. This is the pattern. Again, there's an activator and inhibitor here, activator inhibitor here. So the total space now is four-dimensional. If you just look at the two-dimensional projection in the space of the two activators, the geometry of this is basically that either one activator can be high or the other activator can be high. And so in this case, you can explicitly characterize what the geometry is. And now if you increase the number of cells, if you have 5 cells, now instead of these two stable fixed points that are possible, with one being high and the other being low, there are 5 different stable fixed points. It turns out in this case, 2 of them can be high. This depends a little bit on the parameters I've chosen, but the parameters I've chosen, 2 of them can be high, and the rest are sort of low. And the basic mechanism by which this model works is that you have a homogeneous fixed point, homogeneous in space, which is unstable, and then you flow to a stable pattern state. And what we realized when looking at these discrete systems is that the which maybe should have been obvious in retrospect, is that the unstable manifold from the homogeneous fixed point to the final stable pattern state is actually quite low dimensional for these Turing systems. And so this is locally, for example, you can find out the eigenvalues at the homogeneous fixed point, and you find that most of them are quite negative, and there are only two unstable eigenvalues. So now if you try to look at what are the equations on the unstable manifold, instead of looking at the whole system, on the unstable manifold, it turns out that in fact the equations you can write down as potential. So here, for example, there were these two unstable directions, unstable, 1 and 2. And so you start at you can change variables. So you start at 0, which is the homogeneous fixed point. And then, as I showed you, you have 5 different stable fixed points, stable pattern states that you can go to. And the geometry of that is basically that you start at the center, and then you go to a ring, and there are these 5 different stable fixed points on the ring. And which one you go to depends basically on your initial conditions, which depend on noise. But the interesting thing is that the equations that sort of describe this motion are basically universal in the sense of Landau theory, that you can write down a simple polynomial which captures the geometry of this manifold. So even for this problem of spatial pattern formation, which is now a more complex problem because it's in a high dimensionals, it's high dimensions because there are more number of cells, as well as things going on at the level of the single cell, one can still have some sense in which there is a low dimensional manifold on which the geometry is, or the geometry of the dynamics is still low dimensional. And so this goes back to the question somebody asked of the fly wing. The fact that when you make some heat shock, you get two sets of wings. Then one can think of as the fact that now you can not just think of the Waddington landscape at a single cell level, but really at the level of all of development. There's some few stable phenotypes, at least, and now this is not a precise statement because some mutations can really change things a lot. But there's some sense in which even for many cells you can look at the geometry, which we discovered using this Turing mechanism. So I guess I'm running out of time. So maybe I'll just quickly say that you can also do this now for continuous systems, not just discrete. And these eigen directions that are unstable just turn out to be the Fourier modes that are unstable. And so in the space of the Fourier modes, you can write down sort of universal equations for how the for the geometry of these systems. And finally, you can actually fit things where you can take some Turing simulation which is making a bump, and you can fit it using just the parameters, the 2 or 3 parameters that go into these equations. And so the Turing, again, at the level of the equations, you could put in whatever equations you want, or you could put in random noise and so on. The idea is that the geometry is capturing sort of the behavior that is independent of noise and that is independent of the particular microscopics. Okay, so I mean, this is another example, a 2-dimensional example, a little bit more complex. And again, the idea is that you can take a simple Landau-like theory and fit these systems, which I won't go into too much detail because I'm out of time. But finally, to say that this idea that things are always low-dimensional doesn't always hold. In particular, in this example of bristle patterning in Drosophila, we found that at least for the model that was constructed, it was sort of intrinsically high-dimensional, and there was no low-dimensional manifold for which you can, in which you can reduce the dynamics. And so you really had to make some intrinsically high-dimensional model. But basically, the geometry, in a sense, tells you a lot about the nature of the problem, and ultimately, what are the relevant variables that one should be using to fit any experimental data that exists. So I again won't go through the details, but maybe just quickly I'll say that one is used to the idea of universality in physics. And of course, there's been things from the renormalization group, which now very nicely describe the emergence of universality. I would say this is in biology, it's a much more difficult and unsolved problem. But at least in the context of development, one has a system where one can sort of ask precise questions. There's some, a lot of new experimental data that's available, and there's a history of sort of these conceptual thinking in the field in terms of more emergent description, which makes it, I think, an interesting field to translate some of these ideas into biology. So with that, I think I'll skip this slide and just conclude and say that I've shown you that Waddington's metaphor can be converted to a mathematical statement, and that it's possible to classify these models of self-fate specification into various kinds of geometrical normal forms, which actually have which actually have content in the sense that they're experimentally distinguishable. And finally, that these kinds of models may be a more principled way to fit data rather than microscopic models, which have various parameters, because they capture sort of the essential nature of the dynamics using the fewest possible parameters. And finally, that also in these Turing pattern formation, spatial pattern formation, one can also think of geometry and Waddington landscape. Okay, so with that, I'll conclude. Thank you very much.
Thanks so much. Yeah. So, yeah, are there any questions, comments?
Yeah.
Hey, Archisman.
Hi.
Hi. So, sorry, I'm definitely out. I mean, this out of this field. So let me ask you one probably bit of broad point which I'm missing. So when you say, obviously this any biological system description has to be some way effective, because you just cannot probably go from any microscopic description as such.
Mm-hmm.
But in the context of, let's say for example, when you talk a high energy physics, or maybe this renormalization, so the typical effectiveness is parameterized by this energy in some sense, correct? We write down low energy theory,
Mm-hmm.
and that's the effective descriptions, in some sort of some
Right.
fundamental whatever.
Right.
Now, in that sense, when you are modeling this effectiveness,
Yeah.
so what is that corresponding energy, quote unquote, in that kind of system?
Right. Yeah, I mean, this is a difficult question, and the basic problem is that it's hard. I mean, so you could think instead, in energy, or you can think of length scale. It's the same thing.
Yes.
And it's sort of, I would say, quite an interesting. Biology is quite an interesting system, because a mutation, for example, is in some ways, a microscopic perturbation. At least in some level of some classification of length scales, but a mutation, and this we know, can either have no effect, or it can really change things completely. It can make an organism unviable. And so this means that a local perturbation can either
Yes.
have very long range effects, or it can have no effect, so to say. It seems like. And the nature of the results of the local perturbation are quite heterogeneous in biology, and it seems to me that one is not really used to dealing with heterogeneous systems in physics, because usually you have either you have random systems, or you have then you have some classification of this, or you have homogeneous systems. And I think that's what makes this kind of a description difficult. But I do think ultimately you do have something in terms of length and time scales is quite important. But how exactly one has to do it in actual biological systems, I think, is an open problem.
Okay.
Yeah, I mean, I don't know if that's an answer, but basically, I don't know if there's a good answer to that. But there is no renormalization group for biology, so to say.
Okay.
As far as I know.
Okay, thanks for the comment.
Yeah, thanks, Archisman. That's a really fascinating talk. So we hope we can have you over in person sometime in the near future, when things become a bit more normal.
Yes. Yeah, I would very much like to come, because not only have I not been to ICER, I've actually somehow not even been to Kolkata so far. So.
Okay.
Yeah.
Sure.
Okay, Bhavtosh, shall we end it if
Yeah,
there are
I
no more
guess,
questions.
So if there are no more questions, maybe we could wind up or.
Yeah, thank you once
Okay,
again.
thank you, Archisman.
Okay, thanks a lot.
Yeah, thanks. Yeah.
Thank you.
Okay, yeah.
Bye. Or should I stay back or?
I don't know if, it's okay. I guess we can maybe catch up later, huh?
Yeah, sure.
Thanks so much.
Okay.
Thank you, Archisman. Bye.
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 So somebody has to switch off the recording actually, I think. Bhima or Kamraju's